Okay. And okey-doke. So today's lecture probably is going to be the last lecture about the RNA analysis in general. So we covered part of the RNA analysis during our discussion on the sequencing and the transcription translation process and the cDNA library synthesis. Last week, we discussed about how we analyze one gene by qPCR. And this lecture is going to be how to analyze the bulk RNA or the transcriptome altogether. Probably there will be only a couple of slides left for the next week where we will discuss single cell analysis. So today we are going to discuss a little bit about real-time qPCR, microarrays, and RNA sequencing. And in the next week, probably a couple of slides about single cell analysis. So the progress so far, we know where we are. And just to remind you that there are many ways you can study the one gene at the level of a qPCR. So we are discussing quantitative PCR, but there are other types, which is called digital PCR. Obviously, as you go the more deep you go on the list, the more specialized you have to be. And that's why it's important for you at least to understand very well the qPCR, because you are not going to go to a lab and somebody asks you to make a cDNA synthesis or a microarray prep or RNA sequencing or single cell RNA sequencing. Most probably they will make you work at the level of the quantitative PCR. So you have to know how to design your primers, how you test the primers, and how to analyze the data. And that's pretty much what we covered in the last week. Now, I'm revisiting a little bit the cDNA synthesis, where we discussed that if we want to study the transcriptome and we clone that transcriptome, the first thing is to go to the messenger RNA and reverse transcribe it back into complementary cDNA, which we call cDNA. And if you read through the list of the steps here, we said first we isolate the RNA, then we are generating by reverse transcription the cDNA. And then there is a step here that says generation of the complementary strand. Why is that? Because we want to generate a double-stranded DNA. So we are generating first a hybrid molecule, and then we are generating a double-stranded. So it's important for you here to remember that the cDNA you are using is a complementary to a messenger RNA, but it's yet a double-stranded DNA. And I re-summarized this for you here. When I made a survey after the midterm, some of you mentioned that there were no books added. So there were 3 reference books that were added. You can see that from time to time, I'm visiting some of these books. Sometimes I'm not using books because these things are really new and research papers are better. So you can see that the references are research papers. So this is from that book. Now, when you are generating the complementary cDNA for a library preparation, you would mostly use a primer that binds to the 3' end. And now you know that this primer is the poly T because we have A at the end of the messenger RNA. And then most probably you are generating the hybrid strand, where you get one strand that is a messenger RNA template, and the other strand, it's this guy here, which is the first complementary strand. So this is a hybrid strand that you generate. Now, if you're doing a cDNA library, you have to be careful which primers you are using. And you remember that we can add things to those primers. So at the 5' end and the 3' end of, and the 5' end of the reverse and forward primers. So for example, here, they use BAMH1 and HIND3 added to these green and yellow primers. And therefore, when you generate your first hybrid strand, and then you make the second strand, and now you end up having this complementary double-stranded DNA across the different steps, you can simply cut it with HIND3 and BAMH1, and then you can ligate it to a vector. And now we are speaking about only one messenger RNA. So you can imagine if you have a 3,000 of those, you will have a 3,000 different vectors. Okay, and please, many times I just speak about one molecule because I'm presenting one molecule in the slide. You have to kind of sit back and imagine that this is not only one molecule that we are talking about. So when you are generating a cDNA library, you are taking all RNA. So maybe there's like 20,000, 60,000. I have no idea how many RNA you will be isolating. And from that, you are generating 60,000 or 80,000 of these double-stranded cDNA with different restriction sites on the 5' and the 3' end, and then you are generating this vector. So I think this we designed primers before in assignment 2. Most of you have done it right, and that's how we generate the library. After that, this library, we can sequence it, we can overexpress it, we can do many, many things, but we can do it individually, one by one. We will take this plasmid, sequence it, know the sequence, what is the gene, and study the function. Now, let's go back to the design of the qPCR primers, because last time we discussed that, and we discussed the differences between the regular primer and qPCR primer. The only difference really you have to take care of is that you are using your cDNA, which is the coding part of the cDNA, of the gene. So, and then you have to take into consideration that in order to detect your qPCR, you need factors like a thermocycler that can read the fluorescence and make sure that you are looking at the melting curves to know if your primer works or not. So I'm not going to go in details on this. There are some slides there. In the slide that was describing the primer design, there was a missing couple of words here. So it was written interexonic flanking the entrons. So I just added some corrections. So be careful, go back to that slide and correct if that was not clear for you. So the qPCR primers should be interexonic. Again, it goes from exon 1 to exon 2, or whatever combination you are trying to use. Exon 5 to exon 6 work. Exon 8 to exon 10 work. Depends on the exon you are choosing, but don't make that primer anneal or be complementary to the entron, because then you will pick the genomic DNA, not the coding DNA. And be careful that when you are generating qPCR primers, don't exceed 300 base pairs, and look to the classical parameters that we have to take care of, which is the GC content, melting temperature, and the secondary structures. Okay, now when we are generating the cDNA for the qPCR, then there are two different categories. And your colleague, the last time when he discussed with me on Wednesday, he was confused whether the cDNA is double-stranded or single-stranded. And the one I presented last time, I mentioned that it could be double-stranded. But I have to tell you that there are different types of kits. Like now, if you go to the lab and somebody extracted RNA, so we took some cells and we extracted the messenger RNA from those, we know that the messenger RNA has this poly A tail. So the easiest way for us is to generate the double-stranded hybrid messenger RNA DNA is to have the poly T as a primer. And then we can amplify the cDNA based on the messenger RNA sequence. We could then use the RNase H in that another step where you can degrade that RNA and you end up only with the DNA part. And then you can do a PCR or PCR-like where you use a DNA polymerase to generate your secondary DNA. And then you get to this double-stranded format, okay? Now, be careful. There's something that if you read in molecular biology books, I posted for you here, exactly how it's written in one of the books. It's unfortunate that real-time and reverse transcriptase can both be used as RT in abbreviation, so be careful. So the process of generating a messenger RNA into a cDNA, it's by reverse transcription, right? So some people write RT, so this is the process. Some people also refer to the real-time PCR, which is the quantitative PCR as RT. So that's why I was very careful when I'm putting it on the slides. I call the RT-PCR, not only RT, I call it RT quantitative PCR. And that's how you do the analysis of the gene expression. While the RT in the course, most of the time, till now, I always say RT only for the reverse transcription. I never used RT on its own. So just be careful if you read it, in which context it's going to be, right? Now, we discussed how we generate the cDNA synthesis, and it's important for you to know that there are multiple kits. Some of them we can use directly the messenger RNA as single-stranded, and some we can go to the double-stranded. That is not going to be any change for you in terms of the knowledge of what we have described. If you start with a single-stranded cDNA, this means you would need a couple of cycles more, but you are ending in the same result. So now, when we are generating the cDNA, the first step is the reverse transcription, which will generate this hybrid molecule here in green. And now, the other question your colleague asked is, do we only use the oligo-DT as a primer? The answer is obviously not, because there are other ways to generate the hybrid messenger RNA cDNA. You can use those primers. These are called random hexamers. Should be 6, not 4. They are presented at 4 here. Because those, they bind randomly at different sites of the messenger RNA, and you can obviously convert this messenger RNA into this cDNA. The easiest one and the most efficient and the most widely used is the oligo-DT, because we know that messenger RNA has a tail of multiple A's at the end. So if you were to be asked whether oligo-DT primers or hexamer are used, either one can be used. There's no really preferential between the two. People use oligo-DT more in the labs, probably it's cheaper. And now, when you generate this double-stranded hybrid molecule, you can do nothing, in fact. You can do nothing, and you can just directly go to your qPCR. Because if you take this hybrid molecule and then start doing a qPCR with the specific primers for your gene, say you are working on the gene actin or the gene insulin or whatever gene, you start doing the qPCR cycles, the heat is enough to degrade the messenger RNA. Once you do the first cycle of heating, it's going to be enough to kill your messenger RNA. And you will end up only with this molecule, and you can run your quantitative PCR. Or there are kits that are a little bit more expensive, where in the first step, as I mentioned, you generate this. And then there is a step where you add the RNase H, so it kills that messenger RNA. You end up only with the cDNA as single strand. And then you can add the polymerase at another step, and then you can get the double-stranded DNA. So you can use either this or that in your qPCR reactions. And this is not a question that I will be asking you in an exam. It will not be a question, because this is not the one, you are not the one who decide which one which step you have to stop at, or which kit you're gonna use. Now, if the question is, can you use the cDNA as single or a double-stranded in a qPCR? Yes, that can be a question. But how and which kit we use for a particular experiment, I'm not gonna ask you that. Okay? Now, whenever you generate a hybrid cDNA messenger RNA, that can go and can be used as a template, as is written in this. So this hybrid molecule generated in the first step of RT can be used as a qPCR template, or you can use another kit, as I mentioned, and that will make a double-stranded, and that can be used also in qPCR. It depends about what is your supervisor doing, what genes you are using, what are the reasons if you need to start from a single-stranded or double-stranded DNA. Okay, then we discussed about the types of analysis that we get, which is we normally use the fluorescent dyes in order to detect the amplification. Now, before I go to that, I will stop to ask you if you have any questions about everything we discussed so far. If you have a question, please stop me now. Okay, it seems there's somebody who has. Come on. Question. Question. Jacob. You don't have, okay, just to clarify, you can use single or double strand. Exactly. That's true. That's exactly what this slide says. Both can be used, depends on the kit. And for example, if you come to a lab, all their life they are using oligo-T, they don't want to change that, because if you change from using this type to that type, your experiment might change a little bit. You don't want to add those parameters. And please, guys, be careful that I can't see your chat while I'm presenting, because the slides are taking full. So don't hesitate to speak on the mic if you want to stop me. So then we discussed about the detection, and in the detection process, we mentioned there are two types. And the first one would be the cyber green, which is the cheap one. And I really advise you to see the videos, because that adds a lot of imagination for you instead of just looking of how the slides look. And we mentioned that the TACMAN probes are not only depending on the primers, particularly to the gene, but they have also a probe which binds to that gene. So if this is the gene insulin, then there's a small fragment of DNA that is very specific to the sequence of insulin that is bound by the reporter and the quencher. And only when the two primers generate the PCR product, while generating the PCR product, then the polymerase is gonna break the reporter from the probe, and that's how it becomes far from the quencher, and that's how it becomes fluorescently active. And that's then we can detect the fluorescence accumulation. Obviously, by the end of the cycle of the qPCR, this probe will be totally degraded and will be released out of that DNA. So every time, what happens is, if I would make an example of a cycle for the TACMAN, so there will be the first step, which is the process of 95 degrees where you have the unwinding of the DNA. And then you will start having to drop the temperature when the primer 1 and primer 2 and the probe bind to that DNA. You are amplifying. And then the primer 1 and primer 2 start to polymerase. Once this primer comes here and reach, then it's going to break the probe. The reporter is going to be released, and it will continue all the way. It will remove all the probe, while the other one, it has no issues. It's going to just read all the way. And now you have two molecules starting from one, and you have a fluorescence that is generated. And then you repeat again and again and again. And every time you have the unwinding and then the binding, then those three components will bind again, and the PCR is going to be generated. While on the other side, you have only the primers with the cyber green. There is no probe, nothing, but there are the dye. The dye is only fluorescing when it binds to this double-stranded DNA. So first, it doesn't bind to single-stranded DNA, and the way it's generated to bind to the double-stranded DNA is the same. You have to first unwind the DNA, the primer 1 and primer 2 bind, and then while you are polymerizing and generating the secondary one, then cyber green binds, and then it starts fluorescing. But it's gonna be binding to any generated PCR product. That's why it's less specific. So then this is a summary for you, and there's more information. So again, you start with a double-stranded. You can see here that the fluorescence is inactive only when it binds to double-stranded DNA in condition of cyber. It will become fluorescing, and you're gonna detect those at the end. And there's more details about the TACMan in case there is something not clear for you. And after that, you will start having some period where you don't see any fluorescence, because typically, first few cycles, you don't have enough fluorescence to be detected. And then after that, you will start the amplification curve. That's normally from cycle 13, 14, and up, normally in the qPCR. And we mentioned that a qPCR normally run up to only 40 cycles. Now, this is more details about how the TACMan works, and there's a lot of details for you guys. I'm not going to describe those anymore. Go back in case, just for the primers, in case you have something like this, you directly know that your primer is not okay. It's amplifying one product, two products. So therefore, it's not a good primer. While this is a good primer, here you can see it amplifies only one product. Okay, and then we mentioned that there are two types to quantify the results. And the first thing you look at in a qPCR is if you have a lot of a transcript or a lot of messenger RNA from a gene, then there is a lot of cDNA of that gene. And therefore, this gene in red is abundant. Then you can start detecting the product in cycle 11. While in the green, you already know that it's much less transcript, therefore much less RNA, and therefore much less cDNA that you generated from that RNA, and therefore it needs much longer cycles to be expressed. And now you can directly say that gene red is more abundant or highly expressed compared to gene green. And we use that to analyze. So we have a lot of analysis that comes from this. So it's correlated. The more cDNA you have, the higher the expression. The less cDNA you have, the higher the cycles you need to amplify. The more the DNA you have, the less the cycles you need to amplify. And that's exactly what you see between red and green. And of course, when you do this, you get a curve, and out of the curve, you have an equation, and then you can get, if you don't know what is in yellow, all you need to know is what cycle is that, and then you can amplify, and you know what is the amount of the DNA. So that's pure mathematics here. And this is how we quantify the absolute quantification, or the exact amount of DNA. But as I mentioned in the last lecture, we don't really always do an absolute quantification. And if you want to do absolute quantification, most of the time we need to do digital PCR as well, not only a qPCR. But we can make it as a comparison between two different situations. So you can say that I have cells that come from normal person or a tumor patient, and then I want to check if this gene is up or down regulated. And then you can say, oh, this gene shows up in cycle 15, and this gene appears in cycle 25. So obviously it's more expressed in normal cells compared to tumor. So that's direct correlative or comparison between the two different sets. And the way we calculate it is the delta CT, which is the difference between the cycles, the threshold cycles. It's very easy. As I mentioned before, you will have, forget about this one, you will have a control condition, which is cells from healthy patient, and experimental condition, person who has cancer or something, or treated with a drug. And then you will measure two things at the same time. You will measure your gene of interest and a housekeeping gene, which is S16, actin, GAPDH. Those are genes that do not change. And obviously you can see that in normal condition, the control and the experimental condition, the reference gene didn't change. You can see both here. And then all you need to say, the difference in the cycles in the experimental condition and the difference in the cycles between the target genes, and you can calculate this very easily, taking this calculation. If you type on Google or ChatGPT, because now it's very popular for you guys, you will directly get that equation and you can get the differences. Now, it's important to take into consideration that we are speaking in theory that everything is perfect, but it's not. So if I'm taking an experimental condition where cells are treated with a drug or come from a patient compared to a healthy person, and it's important that we will do things in duplicate or triplicate at least. So let's say we have 3 patients with different tumors and 3 healthy subjects. Then you would always take triplicate of this guy with your gene of interest, whatever that gene is. I'm always saying insulin. Let's say it's insulin or P53 in cancer, P53, and then your reference gene. And the same for the second patient and the same for the third patient. Okay. The same thing we're going to do for the healthy subjects. Healthy subject 1, healthy subject 2, healthy subject 3 with the reference genes. And then we will get the difference from the normal tissue to the reference gene in the normal tissue. So that's the first CT we get. And then we get the difference from the tumor or treated compared to the reference gene. So that's the other CT we get. And then we calculate what we talk about here. So we get the first CT for each of them, this and that. And then we get the delta CT. And that's how you can say, oh, the control gene is less expressed compared to the target gene. So in this situation, if we are talking about cancer, then we will say this gene is more abundant in cancer condition compared to the normal condition. And finally, this was discussed extensively, and I'm assuming that now you guys know what is RIN, and this is very important for the RNA quality. And it is exceptionally important when you are doing bulk RNA sequencing or transcriptomics, because if you have bad quality, it's not worth using. So this is the first step every time you perform RNA-seq or microarray or single cell analysis. You have to always make sure that you have a good quality RNA. So up till now, I hope it's very clear for you now how you perform a quantitative PCR to get messenger RNA from two different conditions, and then you generate them from that messenger RNA or the RNA that you are interested with into a cDNA, whether that is a single or a double-stranded, and then you can run primers that are specific to a gene of interest where you can amplify the product and you can tell, oh, in this condition, I have more or less expression of that gene. So that's the concept. Now, that was only for one gene, okay? Now we are gonna move into the bulk analysis where we can generate data up to 20,000 genes or all the genes in particular tissue or cells in one run. And the first analysis that was able to generate like a widespread analysis, not only one gene, was the microarrays. Then in 2008, we come into the RNA sequencing, and in around 2014-15, we start to have the single cell RNA sequencing. So you can obviously see that we have limitations that we are exceeding from first doing a qPCR on one gene into many hundreds of genes here in the microarray, then to all genes from different cells that are mixed RNA into RNA analysis from each singular cell by itself right now. So now we are at one of the sites that is absolutely amazing in terms of the molecular biology, how much data we can get from very little amount of tissues or very few cells. Now, these technologies based on probes, and if you forgot what is a probe, the probe is a single-stranded sequence of a DNA or RNA that is used to identify a specific sequence, whether that is a DNA or RNA based on what probe you are using. So if you're doing a southern blot, that was a DNA. If you're doing a northern blot, that was an RNA. In particular for this course and these three different techniques, we are using DNA probes. Okay, so don't get confused that you are using RNA sequencing. If I ask you in the exam, in any of these three, are you using RNA probes or DNA probes? Be careful that it's a DNA probe. It doesn't mean that you are using RNA, it's an RNA probe. So, microarrays. So microarrays, these are tools that we can use to differentiate between the gene expression in two or maximum two different conditions. So this is the first limitation we're talking about. You can only get maximum two types of analysis. So the expression pattern is very simple. You will take sample number one, like cancer cells, and number two, which is a normal cell. You will take all the messenger RNA or the RNA in general. And then you would generate cDNAs that are labeled. In this condition here, the cDNA is labeled in red. And in this condition here, the cDNA is labeled with green. Therefore, your cDNA is fluorescing either red or green when it's hit with the light from a specific laser. Now, there is, of course, multiple steps that we are not passing through right now. Probably it's going to come in the next slides. Then after, you're going to mix this cDNA and this cDNA together. You're going to mix them, the green and the red, in the same tube, and then you're going to spot it on different sides of these microarrays. Now, the microarrays, they are similar to a microscope slide, but on top of these slides, as you see these dots here, they are printed with thousands of tiny spots, and each of these spots, we know the position, like 1, 1, 1, 2, we know those positions. And each one of those, it has a cDNA that is specific for a gene. So, for example, now we know that there's 20,000 genes. And if you want to do microarray, then you have spotted those sequences and these dots here. So each one of those dots represent one of the genes. And then the outcome would be, for a particular gene, is it more expressed in the normal cells or in the cancer cells? So obviously, when you mix them together, you have cDNA for gene insulin in normal cells, or P53, and you have from cancer. And the question is, which one is more, the red or the green? And therefore, when you plot them on that microarray, for gene number 1, whatever that gene is, you will get a yellow color, which is a similar mix, green, yellow. So you would say normal cells and cancer cells, they are expressing the same amount of that gene. Now, if you get more green cell, then it's more expressed in the cancer. And if you get more red, it's going to be more expressed in the normal tissue. So that's the concept of the microarray. We have a different number of genes that we know. We have probes for those genes spotted on the microarray. And when we get the cDNA from those two different samples, we label them different colors. And all we get from that is which one expressed more. Okay? So now let's go a little bit more on the steps. So I wrote the steps for you. The messenger RNA molecules are typically collected from 2 conditions. Then these 2 messenger RNA are converted into cDNA. And now you know how we do that. And the cDNA is going to be labeled, one in red and one in green. And after that, the 2 samples are mixed. And then we allow them to hybridize to the microarray. Because now you have a single-stranded cDNA is going to hybridize with the single-stranded that is on the microarray here. And after that, you will check. After the hybridization, you can interpret the results based on the color. And obviously, the color is not what you will get only. You will get also a number that reflects the color. So if it's like more red, it's like 10. If it's less red, it's 9. So the analysis process is not mentioned here. This is only the theory. And after that, you can quickly say there is, like, for example, all these green cells are potentially very high expressed in cancer, while those cells are in red are potentially more expressed in normal cells. So that's a very simple way to analyze it. So if we look more in details into how the microarray spots look like, so you can imagine it here. With these spots, you have a single-stranded cDNA, single-stranded probes that are complementary to a specific gene. This is for a gene 1, gene 2, gene 3, 4, 5, 6, till 9. Each of those is specific for a gene. And then you got your RNA. You generated that RNA into a fluorescent dye and added to it, and then you will perform the hybridization. And after the hybridization, you can do the analysis that we discussed. Now, microarrays can be used in different ways. This is one of the ways we can use them. And nowadays, they are not really very, very popular because it's more like RNA sequencing. So if you have read, or you will see somewhere that microarrays are used, for example, to compare a drug to no drug condition, or compare if there is something changing in the transcriptome of or splice variants or something like that. This is possible. I'm just giving you the most common scenario that microarrays are used with. Okay, and this is one of the examples that also listed to show you that you can predict if, for example, certain type of genes is up or down regulated in certain diseases. And again, I'm giving you the same question so you don't get confused more. It's normal cells versus cancer cells. And the question that they were asking here, we have around 70 genes that we are studying. Each row of those is one gene. Its column, sorry, is one gene. And each row is like a patient. And the question was, which one of those genes are upregulated in condition where the cancer comes back or recurrently come back? So simply speaking, they took a cancer cell from those patients compared to normal cells, and they ran those. And obviously, you can see there are two different populations. The upper population, where you have like this gene here, is mostly not showing up in conditions where the cancer remained for, where the patient was free from cancer for 5 years. While it was more obvious that it comes here in the population where the cancer came back recurrently within the 5 years. So in other words, you can say that these genes are good indicators that if they are upregulated, there is a chance that the cancer comes back faster. Okay? And the other way around, you can read it from this side that these genes in red here, they could be the other way around, meaning that they could show up more in the person who doesn't have the recurrent cancer. So you can make some prediction studies based on this. And again, the microarray is good because you are really targeting a specific number of genes. So on the microarray, you have 70 genes. In other words, you have 70 probes directed to those 70 particular genes. And you are not studying all the RNA, so you don't have noise that comes from all the RNA, the ribosomal RNA, and all that. So the results would be really clear. Okay, I'm gonna stop here with this. There are a few things that, there is this video here that can show you how the steps are made. It's pretty much summarizing the slides. And if you have anything that you didn't get, please either unmute yourself or ask me now so that I can answer your questions. Okay. LoxP site. Sorry, so it's limited to 2 cell types, but we analyze multiple genes. Yes. In this particular slide, there's only 70 genes. I don't 100% recall by heart how many genes you can maximally analyze in microarrays, but it's not really huge. But yes, always you have only 2 chances because it's red and green. And now, again, be careful that when you are doing this type of experiments, you are not only using one cell population. So probably it comes from many patients of the same condition. So when you are doing experiments, you have to always think about duplicates, triplicate. If you are doing a tumor, you will collect from more than one patient, but then it's still tumor, right? And when you have the normal cells or non-cancer, also not only one patient, it's many patients, many healthy subjects, sorry. Okay, any other questions? No questions. So just before moving, you guys can also see now that most of the biotechniques that we were discussing, again, you can start seeing that they come all together now. You can see now that you have to isolate the RNA or the messenger RNA, depends on the type of RNA you need. Then you will need to know how to do the RT to generate the secondary, the complementary cDNA. Then you need to know how to label the DNA in order to have two different populations, red and green. And you can see that this process here is a hybridization, so it's a really sequence specific. So the probe for each of these genes has to be very carefully selected, that it picks only that gene, not other genes. And overall, when you combine all these expertise together, you end up having the microarray analysis. And this is really amazing how molecular biology is evolving. Okay, so with this, we're going to stop discussing about the microarrays, and we're going to move to, if my computer allows, we're going to move to RNA analysis. So one of the limitations we just mentioned is that in microarrays, you cannot study all the conditions that you want. So you are limited in the number of conditions, and you are limited in the number of genes. Because if I go back a step, in order to have a probe for a gene, this means I already know that there is a gene, right? And I already know that this probe is very specific to that gene. So what if I don't know that this gene exists? Then that's the other limitation that we have with the microRNA, because microRNA is a microarray, sorry. The microarray is a known genes. We should know the genes. We should make probes for those genes, and then we hybridize them here in different cells, and then we can study it. But in conditions where we don't know if there's a gene that was transcribed and generate RNA and then the cDNA, then you would never detect it. Or if you detect it, you would not know it. Okay? So that's why RNA sequencing is much more powerful. Because in the RNA sequencing, it's assumingly taking all the RNA population, that some of it is translated to the protein and some not. So in principle, we are taking all the transcriptome. And as I mentioned, the term RNA sequencing the first time came in 2008. And it is one of the most useful tools right now in molecular biology in different labs, where you can study all transcriptome, and you can study the differential gene expression, differential gene splicing, exons, and it's like too many applications that you can get from the RNA sequencing. And the RNA sequencing is similar to what we discussed during the genome lecture, and I think it was lecture number 10, where we discussed the next generation sequencing, how we sequencing the DNA. It's exact same procedure, but there's only one step before the sequencing of the DNA, which is the start material is going to be RNA. So in principle, if that RNA is a gene that generates a protein, then it's better for us to study the messenger RNA. If you are studying ribosomes, then probably you want to do ribosomal RNA. If you are studying non-coding RNA, probably small nuclear or non-coding RNA. That's why RNA sequencing is so powerful. It is not really limited to any type of the RNAs that you're going to use. It's mostly to any RNA exists in the cell. So you can reveal the presence of any RNA and the quantity of that RNA in the whole transcriptome with RNA sequencing. Now, in the few examples I'm going to show you, I'm really focusing on the messenger RNA, but that doesn't mean you can do these experiments on other types of RNA. And be careful, again, in RNA sequencing, you are not really sequencing the messenger RNA because it's just a mediator. We always convert that into cDNA because that's more stable, and that's the one that we sequence It will reflect the messenger RNA, but we are not using the messenger RNA per se. Now, there is a very important step that I put here. You have to be careful that during the preparation of the RNA library, or the whatever that library is, messenger RNA or any type of RNA, then a fragmentation process has to take place. We need, because some of the genes are 1 kilobases, like 1,000 base pairs. Some of them are like 5,000 base pairs. Some of them are like 3,000 base pairs. So how can you make sequencing eventually equal to every one of those genes? The idea is, if you fragment the messenger RNA or the cDNA into molecules that is around 300 base pairs, this is very favorable because then you can sequence those with no problems. So there's a fragmentation step that starts right after RNA isolation. So we first isolate the RNA, either fragment the RNA or fragment the cDNA, depending on which protocol you are using. Both work. For Illumina, most of the time, and this is one of the companies that is very popular, it's between 50 to 300 base pairs in terms of the size. Okay. So also you have to know that it's not only Illumina that is available on the market. There are many big platforms for RNA sequencing. These are very, very expensive. Just to give you an example, if you would like to analyze 6 different samples, 3 from healthy subject and 3 from cancer subjects, like patients. Each sample can cost you $1,000 up to $5,000. So depends on the parameters of the experiment. So it depends on how deep you are gonna sequence, and the length of the sequencing primers you are using and the adapters, whether this is paired reads or no paired reads. I know that these things probably you would never encounter at your level. It's important just to have a reference about it. So when you are designing an experiment, you have to have a good, clear question what you are going to do before going to RNA-seq. Most probably you will have to study something 3-4 years before deciding to go to an RNA sequencing. And then when you're preparing your sample, you have to be careful which RNA you are targeting. If it's messenger RNA, as an example, you would need to use the oligo-DT because we know it's selective for the poly A tail on the messenger RNA. You want to deplete any ribosomal protein RNA because that's too much RNA in the cells. You have to make sure how you prepare your library, whether it's going to be a fragmentation at this step or that step. You have to be careful for the quality of the RNA. The REN number is like really high. And you have to use biological and technical replicates. Like biological and technical replicates, meaning if you are studying cancer, you need at least in cell lines 3 different plates from cancer versus human versus healthy. And if you're using human samples, you need at least 20 samples from healthy subject and 20 samples from patients in order to get conclusive. So there's a lot of parameters you have to be careful for. And after that, deciding on how much the depth of the sequence, because you can imagine that the more depth is needed because some genes are more expressed than others. So the high expressed genes, they don't need depth to be detected. The low expressed genes, you need to sequence more to check those. And such conditions. And finally, you will get tons of bioinformatic data to be analyzed. So you need really high-end computation to do that, and you need really experts in bioinformatics to study these data, because it's not a normal Excel sheet that you will get. You will get an Excel sheet with 20 to 30,000 rows and up to how many conditions you'll study. If you have 20 healthy subjects and 20 patients, this means 40 columns into 20 to 30 thousand rows in that Excel sheet. So you as a human, you can't read that and make comparisons. So in terms of the flow of the RNA sequencing, what do we do? So pretty much now that you know how to do qPCR and microarray, RNA-seq is going to be a little bit more complicated, but it's not that different. So you would need first to isolate. In this particular study in Nature, they were studying the muscular dystrophy and ligaments. So you would isolate first the RNA, and then you will generate a library of the cDNA. So all the cDNA will be generated out. But then when you are preparing the library of the RNA fragments that I said it's going to be fragmented into 50 to 300 base pairs, then you can choose either to tag both ends of the cDNA, and that's called paired ends, or you can choose just to tag only one side of it, which is unpaired end. And in this condition, you are adding adapters. So the cDNA, every cDNA will have molecules added at the end. And these are called adapters. The adapters, I put the definition for you here, are short DNA sequences up to 80 base pairs in length. They are attached to the DNA fragments. Now, in this condition, it's a cDNA double-stranded. And those are complementary to primers. So you can combine them with the primers later on for sequencing and stuff like that. Now, one last time, I'm repeating that fragmentation could happen at the level of RNA or at the level of the cDNA to generate the library. And neither, either of these work. It depends on which platform you are using and what experiment you're doing. And finally, you will have to sequence those guys, and you will get a ton of computational analysis to do. Now, let me quickly describe for you the Illumina processing. So why do we add the adapters? That's very important. So taking all the RNA, generating the double-stranded cDNA is very trivial now for you. You can imagine it's a super easy thing to do, because taking poly A tail as a primer, you will generate all the messenger RNA into cDNA double-stranded in 2 steps. Now, fragment it, you get those small fragments. And after that, I need to analyze every single one of those. So how can I capture all those? In order to capture those, that's what the adapter will help you to do. So first, those adapters will help you to capture each one of these single molecules, because it's random and it happens across all the cDNA, that each of those cDNA get those adapters. And depends on which way you're using, whether paired or unpaired, you at least have an adapter added to each of these cDNA molecules. So this is a very crucial step in the library preparation, which you see here. And in this condition, you have a paired end, because you have 3 different cDNA molecules that we added 2 types of adapters. One that is black, so 80 base pairs on each side that are different sequences. But this sequence is similar to that, right? Now, what will happen is these adapters are your mean to allow binding to the flow cell, And the flow cell, it's simply like a slide that has tons and tons of these adapter binding molecules. So in other words, the adapter is on one side, you add it to the cDNA, and on the other side, it has its complementary on the flow cell. So with this black one, it found the complementary on the flow cell, and that's why it bound here. And this blue one, the same. And this green one, it bind to the other side, which is in gray there. Okay, now there will be an amplification step, which look very weird for you how it happens. But there is a next slide, there's a video that we will watch together. So you amplify those, and you keep amplifying them in a way that you generate a cluster of each. So you generate many of this molecule in red, and then many of this in blue. After many times, you will get a cluster of red, a cluster, all or copies of the same one, copies of the same one. And then you would do sequencing for that. And it's exact same way that we decided, we studied the sequencing before, and therefore you get the sequence of that cDNA, or in other words, that gene, which is a complementary part to a messenger RNA. So then you would say if the blue is more expressed in cancer condition compared to healthy subject, or the other way around. Okay, so that's how it works. I know this is very complicated for you, and that's why I would like to escape from here, and let's watch this video together. Oh, wait, one more thing. Before I go to this one, you still see my screen, I hope. So the first step, you took the RNA and you make this double-stranded cDNA. Then you added the adapters, 80 base pairs on each side. I'm not gonna ask you how did you add those? It's more complicated than what you think. But we added the adapters, but these adapters are complementary to primers. This primer here in green is complementary to the adapter in green, so it can bind to it. And there's another primer in pink that is complementary to the adapter that is added to the cDNA. And now this is one cDNA molecule. Imagine you have 50,000 of those, so you will have 50,000 adapters that are added. 50,000. You have only two types of adapters, huh? Two types of sequences, green and pink, added to the 50,000 cDNA, and then you have primers that are complementary to those. So if we run one PCR cycle, only one PCR cycle, so where this primer binds here and amplify that all the way to the end, and this primer here bind and amplify all the way to the end, you will end up having this molecule, which has cDNA, primers, and additional molecules that were added to the primers. One of them is called index. So that's how we know one cDNA from another. And you have this small part that allows binding to the flow cell. So let's watch this video together. And probably if you have questions, you can ask me later on, okay?
Sample preparation begins with extracted and purified DNA. The first step in Nextera sample preparation is tagmentation. During tagmentation, transposons simultaneously fragment and tag the input DNA with adapters. Once the adapter...
So you hear he already said, this tagmentation process is you cleaved or you digested your RNA into molecules around 50 to 300 base pairs, And at the same time, you added 80 base pairs adapters on both sides of the cDNA. And then
have been ligated. Reduced cycle amplification adds additional motifs, such as.
So the next step was those primers that are complementary to the adapters. But the primers, you can add more molecules to the side, exactly like you added restriction sites in your assignment. This sequence can be specific to that cDNA only. And this green part and yellow part, it allows the cDNA to stick to the flow cell. So that is not floating anymore. It will be binding to the flow cell.
The sequencing primer binding sites, indices, and regions that are complementary to the flow cell oligos. Clustering is a process wherein each fragment molecule is isothermally amplified.
So that's the flow cell. The flow cell is simply a slide that has lanes, and it has millions of those adapter molecules, complementary parts of to the adapter molecules already hybridized to those cells. So you will see that there's only two types of complementary adapters.
The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface.
I'm going to stop here. So now, when they generated Illumina, when they generated these oligos that are bound to the flow cell, they know that there's 80 base pairs they will use for adapter. They will know that there's like 20 base pair or like 510 base pairs for sequence and enzyme, and part of it that is complementary to this guy there or that guy. And therefore, you can see that there was a transition in this video that if you go back 1 second.
Each lane is a channel coated with a lawn composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos.
So what I wanted to say is that the sequence of the yellow and the green, we already know when we design our primers, index, and adapter, it's complementary to those two guys. So the Illumina knows that from the beginning. And that's how in the video when he is going forward, this will be kind of transition into yellow or green.
Oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away.
So this is the first important step, because now you have a molecule that is attached to the cell. Before, your RNA molecule was floating all the time. But with this first hybridization step and removing that part that flow away, you got the first strand that is stripped to the flow cell.
Strands are clonally amplified through bridge amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions.
So this is for only one cDNA out of the 100,000s you have. And then you're going to repeat it.
Resulting in.
So this cluster is different from that, different from that, different from all. So there is around 144 million that you can read in an experiment like this. You can read at least 20 million up to 144 million of those in an experiment.
Clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands.
So now you remember when we do the sequencing, you know that we need to do a primer that binds, and then the fluorescent DNA will come and bind, right? So These steps that are happening, these are not trivial. There are a lot of changes that Illumina needs you to do in terms of the experiment. So like to wash this out, there is a step that is needed. And now we are going to have a single-stranded that we can sequence, right? So he's going to show you the
The three
sequencing.
prime ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer
Remember that we had two primers, one forward and one reverse, and they had different colors. So now we are sequencing the first round.
to produce the first read. With each cycle, 4 fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determine the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read.
So this is particularly important to know that if you have paired ends, then now you are reading again the second strand. If you have only single strand, you will not read it.
The read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' end of the template is deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Index 2 read product is washed off at the completion of this step. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends blocked. The original forward strand is cleaved off and washed away, leaving the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is washed away. This entire process generates billions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments.
Okay. So that is absolutely complicated for you, I know. But it's important to show you how things we are speaking in a very... Now you can understand my pain in describing those things for you, to be honest. The thing is, I'm not going to ask you how Illumina itself works. It's important for you to visualize and imagine how things work, but I'm not going to ask you how the steps of Illumina video that I just showed you. And in other words, that shows when you're doing an experiment, there are too many steps that if you try to explain, they cannot make so much sense because it's so difficult just saying you do this and you do this if you don't see it by eye or you do it by hand. Okay, so with this we end up with just one last slide showing that this is one of many applications of the RNA-seq. So the only disadvantage of the RNA-seq from too many cells is that if you take these cells from one patient and the same amount of cells from the other patient, you are getting the average RNA from that and the average RNA from the different patients. So this is not an absolute number, could be differences that you can miss. But you can, of course, use RNA sequencing to identify novel transcript that you have never studied, messenger RNA that you have never studied. You can measure the difference in transcript between two different groups, like cancer versus healthy. You can identify novel splice variants, and that's how most of the splice variant we know. And probably with RNA sequencing, you can annotate better transcripts. You can revisit some genes that you think it's 3 exons, 4 introns, and 4 exons, 3 introns. Probably you can have more than one annotation for this. So there are many, many applications. There are some written for you here, and there's a text explaining it. And with that, we are stopping this session, and I will stop to ask to get any questions, because I'm assuming there will be some. Oh yes. So will the steps of cell processing be? Oh my God. So there's one person, his name is Loxp, and the other one, his name is Cree Recombinase. You guys are very good in asking questions related to exam. I'm not gonna ask you to describe steps of the Illumina, because as I said before, Illumina is one of the platforms. There are many other platforms. Plus, you will never yourself or any researcher in lab perform this step, because you will outsource it to Illumina. So that's not something I will ask you. But the details of how you prepare the cDNA and how you fragment the cDNA and the type of the cDNA, or the steps, or what are the multiple steps of the RNA sequence, that's probably where I will ask you questions. Okay. So sharing. Could you recap conditional normal knockout procedures for assignment 3? Okay, so so that will be really long to recap right now, because I need to have slides. But if you can re-specify your question, could you please re-specify your question better so I can answer better? Conditional knockouts. So conditional knockouts, there are 2 types. Just give me 1 second.